Welcome back, friends. In previous video we discussed about the aldoses and the ketoses, and in this video I want to discuss with you about the facts about disaccharides. Now disaccharides, they are sugars formed as the result of the combination of two monosaccharide sugar. The term suggests di means two and saccharides comes from the word monosaccharides. So disaccharide inatengenezwa pale ambapo monosaccharides mbili zitaungana. Mara nyingi huwa monosaccharides ambazo zinaungana zinakuwa ni hexoses, na reaction ambayo inaunganisha hizo disaccharide mbili inaitwa condensation reaction. Now what is condensation reaction? We normally define it as the reaction which involves the removal of water molecule from reacting molecules. So what we need in the condensation reaction is two reacting molecules and the removal of water molecule from those two reacting molecules. So for example, as you can see in the reaction here, we have two glucose molecules and we have two hexose molecules, and in the condensation reaction we remove water to form a disaccharide. This is a disaccharide, and in hydrolysis reaction the backward reaction is hydrolysis, which involves addition of water. So during condensation reaction a bond is formed by the removal of water molecule. Now we are saying that the backward reaction that involves addition of water molecule to break up disaccharide is known as hydrolysis reaction. Kwa tunapokuwa tunalowesha maji, tunafanya hydrolysis, tunakuwa tunalowesha maji ili kuweza ku break down ile disaccharide kwenye monosaccharide. Hydrolysis is normally done by the enzyme kwenye organism. Kwa ndani ya mwili kuna enzyme kama amylase, salivary amylase inafanya hydrolysis ya kutoka starch kwenda kwenye maltose. Na tutaenda kuona baadaye kwenye hizi disaccharide kwamba disaccharide zenyewe ndio zinaweza kaungana zikatengeneza polysaccharide. Kwa maana yake tukitoka kwenye polysaccharide tuna digest kwenda kwenye disaccharide, baada hapo tuna digest kurudi kwenye monosaccharide. So in the body they are formed by the hydrolysis reaction are catalyzed by enzymes, but in laboratory we use the dilute acids such as HCl, and we see this when you are doing the practicals of reducing and non-reducing sugars in the laboratory. So the bond formed between the two monosaccharides as a result of condensation is called glycosidic bond. This is very important to remember. In a carbohydrate, the bond joining two carbohydrate residue together, it is called glycosidic bond. Very important to remember, glycosidic bond. And we have two types of glycosidic bond depending on the carbon atoms which have been joined. If the first carbon of one molecule join with the fourth carbon of another molecule, that means, for example, if this is my glucose molecule, and then here the first, the second, the third, the fourth, the fifth, and then if we draw another glucose molecule here, and this is my first, my second, my third, my fourth, my fifth, and then as you know the sixth carbon is outside the ring. So what I mean here is that assuming here we have hydroxyl, then hydrogen, and here we have, assuming here we have hydroxyl, then hydrogen, hydroxyl, then hydrogen, and then here we have hydrogen, hydroxyl. So what I mean here is that during bond formation, hydroxyl of this molecule can take oxygen from that molecule. And what will happen here, as you can see, this is the first, this is the second molecule. So what will happen is that we'll end up with a molecule looking like this. The first, the second, the third, the fourth, the fifth, then the sixth of the first molecule. And then here we will have a bond. You'll have a bond formed here. A bond formed by the removal of water. Removal of water will form a glycosidic bond. Hydrogen, then a glycosidic bond. Hydrogen, and here we'll have hydroxyl, then hydrogen. 1, 2, 3, 4, 5. So what I meant here is that this is the first carbon, then this is the first carbon of another molecule. That's why it is called 1,4 glycosidic. And sometimes for some of the molecules, for example, when glucose combines with fructose, they form 1,2 glycosidic. Or in cases of branching, because normally branching occur at this carbon, so they form 1,6 glycosidic, as we shall discuss later. But branching occurred this carbon normally, that's why it is 1,6. That is the first carbon combined with the sixth carbon. So the bond is called glycosidic bond, and is normally forms between the carbon atom 1 and 4 of neighboring units, A1, 4 or 1,4 linkage, 1,4 bond or 1,4 linkage. The process can be repeated many times to build up the giant molecule of polysaccharide. So if it takes place only one times, that condensation reaction results into disaccharide. But when it repeated, it can form giant molecule of polysaccharide. The monosaccharide units are called residue once they have been linked. Thus, a maltose molecule contains 2 glucose residue. The most common disaccharides are maltose, in which here maltose is the result of combination of 2 alpha glucose. Then we have lactose, which is the result of combination of glucose and the galactose. And then we have sucrose, which is the combination of glucose and fructose. So let's discuss one disaccharide after another. Let's start with maltose. Maltose, it occurs mainly in breakdown products during digestion of starch by the enzymes in the body during digestion. So by the enzyme amylase in the saliva or in the pancreatic juice, amylase catalyzing the digestion of starch into maltose. So maltose is the disaccharide. This commonly occur in animal and in germinating seeds. So in germinating seeds, the later germinating seeds is made in brewing beer. Po katika mbegu zinazoota, hii maltose ndio inatumika katika kutengeneza bia. Katika barley grains, zile nafaka zinazoota kama mahindi au kama ngano, wanaweza kuzitumia kama source ya starch. Na pia hata kule kijijini ukienda unakuta pombe za kienyeji, wanakuwa wanachukua yale mahindi wanayaloweka, baada hapo yanavoota wanachukua yale mahindi wanaenda wanayanika wanayasaga. Kwa wao hawaelewi kile wanachokifanya ni nini, lakini kule kuyaloweka na kuyaruhusu yaote, ile pale wanakuwa na convert toka starch kwenda kwenye maltose, ili ile maltose waweze kuifanyia brewing iwe pombe, sasa iwe alcohol, and that process is called malting. So germination of barley is stimulated, and this requires in the conversion of starch to maltose, a process known as malting. So malting is the conversion of starch to maltose. The maltose is then fermented by yeast to alcohol. So there in villages hawaelewi kwamba wanafanya malting, lakini wewe ambaye unajua, kurudi kama kwenda kijijini waambie pale tunafanyaga malting. Tell them that we are doing malting. So maltose is formed when 2 alpha glucose units combine with the removal of 1 water molecule. Kwa tuna 2 alpha glucose molecules na combine, tuna remove water molecule kuform 1,4 glycosidic bond. Na kama ambavyo unaweza kaona hapo, unaweza kaona ili kwa imechorwa na penseli, kwa hiyo this can be more precious as compared to this, but it's the same thing. So we have this alpha glucose molecule, and then we have this alpha glucose molecule, and this is our first molecule. Our second molecule is here. So on adding them, we must release 1 molecule of water. So the hydroxyl group from this carbon atom 4 of the second alpha glucose molecule combine with the hydrogen group to form water. So in condensation reaction we form 1,4 glycosidic bond. This is the bond which can be called as 1,4 glycosidic bond because it is formed between the carbon atom 1 and the carbon atom 4 of the neighboring atoms. So this is the maltose molecule. If you're asked to draw, you may draw like this. So sometimes maybe you may say you don't understand well how you have drawn that. The actual structure was supposed to be like this. So the line which was drawn here below this is not present. Kwa structure yetu itakiwa na mchoro wa pencil. So when maltose is formed, the hydrogen from carbon atom 1 of one molecule combined with the hydroxide from carbon atom 4 of another molecule to form water and leave a 1,4 glycosidic bond. Kwa hiyo baada hapo tuende moja kwa moja kwenye lactose. Kama ambavyo umeweza kuona, maltose ndio ambayo inapatikana kwenye mahindi au mbegu wanazotengenezea pombe za kienyeji au hata pombe za viwandani. So another molecule is lactose. Lactose is called milk sugar kwa sababu inapatikana kwenye maziwa na ni important energy source kwenye young mammals, yani wanyama ambao ni wadogo hasa wanaonyonya. Hiyo ni kwa binadamu kwa sababu wanatumia maziwa. Kwa maziwa mle ndani kilichopo mle ni lactose. Kwa lactose, it can only be digested slowly, so gives slow steady release of energy. Kwa unaona hiyo raha yake, maziwa yanakuwa digested pole pole. Kwa mtu anakuwa anapata ile energy muda mrefu kuliko ambaye angeweza kula carbohydrate yenyewe. Lactose is formed when alpha glucose combined with galactose is shown below. So any combination ya alpha glucose na galactose na reaction kama inavyoonekana hapa. Inaonekana hii ni alpha glucose, hii ni galactose. So the difference between alpha glucose and the galactose is the position of hydroxyl group in carbon atom 1 and carbon atom 4. And as you can see here, this hydroxyl combined with hydrogen, and in the condensation reaction the bond formed is like this one, and this is called the 1,4 glycosidic bond. As you can see in the information below, that the major difference between the glucose and the galactose, the position for hydroxyl group in carbon number 4. In carbon number 4. So this is the major difference between beta glucose. And galactose. Major difference between beta glucose and the galactose. So when it is below the ring, then the sugar is glucose. When it is above the ring, the sugar is galactose. Lactose is the reducing sugar because it has free aldehyde group. So as you can see here, what we are calling as the free aldehyde group is this one, because it does not involve direct in the bond formation as this one. So this is our free aldehyde group, making lactose a reducing sugar, as we shall discuss later. But our last disaccharide is sucrose. So sucrose is also called cane sugar, and it's found in sugarcane. It is the most abundant disaccharide in nature. It is commonly found in plants, where it is transported in large quantities through phloem tissue. So tunavyoona planti inasafirisha vitu toka kwenye majani kwenda kwenye mizizi. Vile vitu vinakuwa vinapita kwenye phloem kwenye form ya sucrose. Mostly hata kama kule plant ina form triphosphate, lakini na convert kwenda kwenye sucrose. So it make a good transport sugar because it is very soluble and can therefore be moved efficiently in high concentration. Kwanza ni good transport sugar kwa sababu it is very soluble. Lakini pili, it is also relatively unreactive chemical. Kwa ukiulizwa why sucrose is the good transport sugar? First, because it is very soluble. Second, it is relatively unreactive chemical. This means it tends not to enter into general metabolism on its way from one place to another place. Kwa inavyotoka sehemu moja kwenda sehemu nyingine ya mmea, inakuwa haingii kwenye metabolism. It is sometimes stored for the same reason. Kwa hiyo inahifadhiwa pia muda mwingine. Kwa sababu gani? Kwa sababu yenyewe ni unreactive. It is obtained commercially from sugarcane and sugar beet and is the sugar we normally buy in shop. Kwa sukari ambayo unaenda kununua kila siku dukani yenyewe ni sucrose. Now sucrose is formed when alpha glucose combined with fructose is shown below. Alpha glucose combined with fructose is shown below. Now sucrose is the non-reducing sugar. Sucrose is the non-reducing sugar. Why? This is because both aldehyde group from the glucose and the keto group from fructose are involved in bond formation. Therefore, there is no free reducing group in sucrose molecule. As you can see, this is our aldehyde group and this is our keto group. All of these they used in bond formation to form the 1,2 glycosidic bond. The bond in the sucrose is 1,2, not 1,4 as we know. And this question of why sucrose is the non-reducing sugar is normally asked in exams. So I beg you to be very serious to remember these points in your exams. And now let's look physical properties of disaccharides. They are small molecule, they are sweet, they are readily soluble in water, they are crystalline in nature. So the properties of disaccharides, they are similar to those of monosaccharides, but the chemical properties, they cannot reduce Benedictine solution. So disaccharides, they are non-reducing sugar, except maltose and lactose. So maltose and lactose, they are reducing sugars. The second thing, or the second chemical property, they form a storage product which is sucrose in organism and can be hydrolyzed into constituents, monosaccharides, and they are used as respiratory substrates. So sometimes they form the storage molecules and they can be stored or hydrolyzed when they are used. They can be hydrolyzed to form simple substance called monosaccharides. So simply here, the chemical properties is this, the first one, and this, the second one. First is that they are non-reducing, except sucrose and maltose. And second, they can be hydrolyzed to form simple substance. And in case of function, sucrose is the main form in which organic salts they translocate. And the second function is about the storage function. Now, let's see about the test for reducing sugar. In the laboratory, when we are testing for reducing sugar, we are using Benedictine solution. Now, Benedictine solution contains copper 2 ion. So, reducing sugar, they have the ability to reduce this by addition of electron into copper 1 ion. Copper 2 ion appears blue, and copper 1 ion appears brick red. So, any sugar which has ability to reduce this by the action of electron into that one, the resulting solution or suspension will appear as brick red. All monosaccharide and some disaccharides such as maltose and lactose are reducing sugar, means that they can carry out a type of reaction called reduction. Sucrose is the only common non-reducing sugar. Only common non-reducing sugar is sucrose. Two common tests for reducing sugar make use of a bit of this sugar to reduce copper from a valence of 2 to valence of 1. Both tests, one among them is Benedictine test and another is Fehling test, involve the use of alkaline solution of copper 2 sulfate, which is reduced to insoluble copper 1 oxide. So this comes from copper 2 sulfate, and then this forms copper 1 oxide. So copper 1 oxide, the copper will appear as brick red. And we shall see later about the basis of this test and different ways to answer the question which can be asked you. Now subscribe my channel. The next video we shall discuss about the polysaccharide. We shall start polysaccharides. So subscribe my channel and share the link to your fellows and tell them to subscribe for more updates. Thank you and have nice studies.